everyone, I am Khawla. I'm a PhD student at Laval University, and today I'm gonna talk on the application of deep learning to small mass spectrometry data classification. So let me start first with our research context. Our team is working on the improvement of clinical diagnosis in two different contexts. The first one is the cancer diagnosis. In this context, the most common practice is the use of biopsy followed by anatomopathological examination. But this practice has a lot of disadvantages since it's time-consuming, it requires the presence of skilled pathologists, and is unsuitable for large-scale analysis. The second context is the infection diagnosis. The most common practice is the biochemical evaluation, but here also this practice is not adapted for rapid analysis because it requires bacterial culturing steps before. So there is a clinical need to develop new accurate, rapid, and non-invasive tools. Mass spectrometry is perfect for such purposes, since it provides no-targeted molecular information on the millisecond time scales. The particles analyzed by a spectrometer will be separated according to their masses, which results in a molecular signature for each analyzed tissue. Recent advances in MS profiling enable its use for real-time cancer diagnosis without the need for biopsy, and also for real-time infection diagnosis. But before using MS technology for these clinical applications, we need first to develop efficient classification models. Our motivation for this study comes from the fact that many real-world applications do not have access to large datasets because of data scarcity. In medicine, it is often the case that some samples are only accessible in limited amounts, especially if we are working on a rare disease, which complicates the design of efficient classification models. So our objective for this work was to develop a rapid classification pipeline for cancer and infection diagnosis on small 1D MS data by machine learning approaches. Here is the standard MS data classification pipeline with conventional machine learning algorithms such as support vector machine or random forest. Raw data must undergo pre-processing steps followed by a feature reduction phase to avoid the curse of dimensionality before arriving at the machine learning analysis. This pre-processing involves several interdependent steps, but the main limitation with pre-processing is that it is time-consuming. There are no golden standards for the optimal choice of parameters at each individual step, for their quality evaluation, and for the best steps combination. Plus, a pre-processing can be effective on a given dataset, but not on another dataset generated with another instrument. So all these factors lead to the fact that the standard MS pipeline for classification is not adapted to a rapid analysis, while our proposed approach is to use raw data directly for the machine learning analysis step. One of the algorithms able to do this are the convolutional neural networks, or CNNs, because they offer various advantages over conventional algorithms, such as being efficient on raw data, which discards the need for pre-processing before the classification. The feature selection step is integrated with the classification, and maybe the most important property is the robustness to spatial variation, because not all the peaks will show up at the same MZ location, and not all of them will have the same level of intensity across spectra. So all this technical and biological variability in MS data will be taken into account by CNNs during the classification. CNNs are designed to learn a representation from an input signal with different levels of abstraction. The input data will go through two types of layers: the convolutional layers for the feature extraction part and the fully connected layers for the class decision part. Here are our small clinical MS datasets. The first one is a canine sarcoma dataset, which contains 1 healthy and 11 different sarcoma types. The second one is a human ovary dataset, which contains a healthy and a cancer class, and the third one is a microorganism dataset, for which we considered first a 3-class classification to distinguish between a gram-positive bacteria, gram-negative bacteria and yeast, and then a 5-class classification to identify each of the strains. So you notice that these datasets were generated in different contexts. The canine sarcoma and the human ovary are in the cancer context. The microorganism is in the infection context. But also they were generated with different organisms, with different instruments, with different ionization sources at different resolution, etc. So here are the result of classification with the CNNs. So for simplicity, I'm showing only the global accuracy metric, but CNNs performed poorly for all the classification tasks, which was expected due to the low number of samples. So to complete the picture of CNNs, because at the beginning I talked about their advantages, but as each model they also have limitation, and their main limitation is that they are data hungry, which means that they need to be trained on very large data sets to be efficient. So our alternative was to test transfer learning. Transfer learning is a two-step process. A model is first trained on a dataset containing a large amount of data to extract an accurate representation. This representation is then reused to build a new model based on a small dataset. So to apply the transfer learning, we use a dataset accurate in another biological context, because the purpose of the study was to analyze the difference between white and gray matter in rat brain, but it was the largest dataset available for this study. Here is the schematic representation of transfer learning on our MS datasets. The feature extraction layers, hyperparameters, are pre-trained on the rat brain dataset, and the fully connected layers, hyperparameters, are fine-tuned on the 3 small clinical datasets we want to classify: the canine sarcoma, the microorganisms, and the human ovary. So here are the results of the transfer learning on our datasets. We have sizable gains in accuracy were obtained for all datasets compared to CNNs from scratch, which means that training a model with extracted feature even from an unrelated field is better than training the model from scratch when the dataset is small. The 3 and 5 class classification microorganisms were accurate, but we still need some improvement for the cancer datasets, the canine sarcoma and the human ovary. So instead of doing a transfer from one source to one target dataset, what we propose to improve the classification is a new approach we called cumulative learning, which consists in training a CNN model on a first dataset, then on a second, on a third, etc., until arriving to the target dataset. For this, we used the rat brain dataset I presented previously, the microorganism, because we obtained an accurate representation for this dataset with the transfer learning. We also used a beef liver dataset and the second human ovary dataset. So here is the schematic representation of cumulative learning on our datasets. The CNN model was first pre-trained on the rat brain, then on the beef liver, then on the microorganism, before fine-tuning its hyperparameters on the canine sarcoma dataset. For the human ovary dataset, we trained first the model on the rat brain, then on the second human ovary, before fine-tuning the hyperparameters on the ovary dataset we want to classify. So cumulative learning improved classification accuracy for both datasets, even for a low resolution dataset, as it is the case for the human ovary, and for heterogeneous dataset, as it is the case for the canine sarcoma, because for some classes samples were gathered from dogs at different ages and from different breeds. So to conclude this presentation, CNNs offer an end-to-end solution since they are efficient on raw MS data, which opens the door to develop rapid and maybe real-time applications in the clinical domain. We showed for the first time that we can obtain an improvement of classification through successive phases of training, which is an alternative when the number of samples is small. The principles demonstrated in this work could be beneficial to other domains where training samples are scarce. We showed also the diversity of tasks between datasets that didn't appear to share common features because the rat brain is a lipid dataset and the human ovary is a protein dataset. So we start to think out of the box because the model will not search for a specific feature in the source dataset and look for it in the target dataset. It is more a question of general landscape data presentation. Among the novelties, we showed that the model was trained to predict only two classes, the white and gray matter in rat brain, and was efficient for classification in 2, 3, 5, and even 12 classes. And the last point is when we did the transfer from dataset 1 to dataset 2, we obtained very good accuracy. When we did the transfer from dataset 2 to 3, we obtained also a good accuracy. So when we tested the final model on the datasets used during the training, the rat brain, the beef liver, and the microorganisms, we found that the model accumulated MS knowledge through the several training phases without any losses. We are a little bit ambitious, but we think that maybe a generic representation of MS classification problems may exist because a single model could be effective on different tasks. So just to finish, thanks to all my lab members, my supervisor Arnaud Droit, and all our collaborators from the University of Nice and the University of Lille in France. And thank you for listening.